good evening. Do please sit down. For those of you who don't know me, I'm Tim O'Shea, Principal and Vice Chancellor of the University of Edinburgh. Tremendous pleasure to welcome you to Professor Mark Stevens' inaugural lecture entitled Confronting the Microbial Menace in Our Food. I'm particularly pleased to welcome in the audience Mark's parents, Sue and Peter Stevens, and his wife Jo and son Ben. Very, very neat. So we've got the family grouping there. I'm also delighted to welcome 4 school students from Queensferry High, Broughton High, James Gillespie High, and you, the school students, and the rest of you that aren't in the front 3 rows. What you need to do is listen very carefully to Mark, because becoming a high Hegian professor, particularly at somewhere like the University of Edinburgh, is desperately hard to do, and he will demonstrate the art that got him there. So pay attention. He was educated at the University in Warwick and Leicester, worked for a while in Compton, came to us in 2011, quickly got a personal chair, then was appointed research director of Roslin, and is now also one of the deputy directors of Roslin. His very successful career marked, and the markers of a successful career for a high Hegian professor are people give you lots of money to do important stuff. He's had the major grants. You supervise lots of PhD students, because that's the next cohort, and that's the extremely important thing, and he's been very effective at that. You publish. The University of Edinburgh puts particular stress on public understanding, public outreach, and Mark has been exemplary at that, and he gives international lectures. So those are all the very good, those are important, those are the esteemed markers, and any of these creatures here in the red gowns in the front, they were, if you looked at their CVs, you'd find those esteemed markers. But why does what he does matter? Well, he matters because he's looking at foodborne pathologies. These cross the spectrum of One Health, and it is really very important. These pathogens kill lots and lots of people who eat the wrong bit of chicken or the wrong bit of lamb or whatever. So this is very, very important work to be doing. It contributes, doesn't just contribute to Scotland, it contributes to the world. And some of his work is on chickens, and I'm very proud to know that, which I learnt from the Roslin, that there are 7 billion people in the world, but there are 50 billion chickens. But some of that chicken protein isn't that good, or it gets bad. And things like that mountain of chicken protein that is killing some people, it is vital that people do the sort of research Mark does, so please welcome him.
Principal, head of college, director, learned colleagues, family, school children and members of the public, thank you so much for coming to my inaugural lecture. It's lovely to see so many of you here at the end of a long Monday. So I'm Mark Stevens. I want to talk about food poisoning this evening, which is always a dreadful topic just as your thoughts are turning towards your evening meal. But before I tell you a little bit about the research that I and my laboratory have done over the years, I thought you might like to know a little bit about how I came to be personal chair of microbial pathogenesis, because I wasn't always a professor. Back in the day, I've kept penguins, and this is me, age 3. I've absolutely no idea what I've got down my jumper here, but I'm sure it was delicious. I've done a bit of swimwear modeling and tried my hand at acting. This is me, age 6, as a pirate. I've done a fair bit of rock climbing and also spent a lot of time at theme parks. And for clarity, that's me on the left. So it was around about this age, at 18, with a handful of mediocre A-levels, that I was able to go to the University of Warwick to study microbiology and virology. And I was really excited to do that because at this time, you could study microbiology, but usually as part of a degree in biological sciences, with some specialism in the third year of study. But this was a degree course where right from the outset I could study microbes and viruses. And I had grown up in Leicestershire and made it as far as the University of Warwick, which is about 30 miles away from home. And I think it's fair to say that at least initially, the University of Warwick and I really didn't get on. So I stayed here at Cryfield Halls of Residence. It was built to the same designs as a Swedish open prison. And for me, it very much did feel like that. I really didn't settle at the University of Warwick and felt desperately homesick in the first term. By the middle of the second term, it was pretty clear that something was going to give, and it was pretty close to being me. I was so desperately homesick, and a very timely intervention at that point from my mum and dad really saved my academic career, because I was ready to give it up. And they bought a small second-hand car so that I could travel from home to the University of Warwick. And I did that every day for the remaining 2.5 years of my degree. And without their intervention, there's no doubt I wouldn't be here as chair of microbial pathogenesis today. So it was a stable platform from which I could launch myself into my undergraduate studies. And for those school pupils in the audience, take comfort here, because I was among the lowest intake in my course in terms of my A-level grades, but I managed to come out with a first-class honours degree. So if you don't quite get the grades that you want right now, don't worry, it's not a barrier to academic success. With application, you can absolutely get there. So I graduated in 1992 and quickly took up doctoral studies at the University of Leicester. So I'm still living at home at this point, and now I made it as far as the University of Leicester for my PhD studies. So here I'm traveling just 8 miles a day to study the regulation of capsule expression in pathogenic E. coli. And this was with Professor Ian Roberts, who at the time was a Lister Institute Research Fellow. He's now Research Dean at the University of Manchester. And it was in my time here that I produced my first significant paper, hard to believe, already 20 years ago. This related to how this particular type of pathogenic E. coli, which cause meningitis in neonates, express this sugar coat on their surface. We call this a capsule, and it's vitally important for these bacteria to evade host immune defenses. And we studied some of the molecular detail of how the genes encoding this sugar coat come to be expressed. I identified an important regulator of this process and went on to show how it acted at a molecular level. I won't go into the detail. Suffice to say, it causes the enzyme that transcribes our DNA into RNA transcripts, which are then read into proteins. It causes that enzyme to run a red light, and so it doesn't stop at stop signals. And this is how some of the critical genes involved in capsule biosynthesis come to be expressed. And this phenomenon of so-called transcription anti-termination was rarely understood and found in bacteria at the time. So this is graduation day, and it's hard to believe that in the very same robes, 20 years later, with rather more grey hair, wrinkles, and miles on the clock, I'm here today. So after my PhD studies, I took a break from working with bacteria and went to work with Professor Wendy Barclay, who's now a distinguished influenza virologist at Imperial College London. And I spent 3 years working with Wendy on how influenza viruses express their genome and make more copies of it in order to produce new influenza viruses. And I had a happy spell, having finally moved away from home. I'm now down at Reading at the University of Reading. And it was here that I met my wife, Jo, who's in the audience. She was a PhD student at the time there that I was a postdoc. And we were married in 2002, and in 2004 came along our son, Ben. So, still living in Reading, I moved across to the Institute for Animal Health when an opportunity came up for me to return to working on pathogenic E. coli. And I did this initially with Dr. Tim Wallace, who was an absolutely fantastic mentor to me. He really taught me what we mean by the term pathogenesis, the production of disease. And I not only studied E. coli 157 in cattle, but developed interests in Salmonella in cows, pigs and chickens, later in Campylobacter, and on some other microbes that I'm not really going to speak about today. Now, it was with much sadness in 2011 that a well-functioning team of around 11 people and myself were dispersed through a process of redundancy when the Institute for Animal Health became the Pirbright Institute, now centred in Surrey, and all the work there on bacteria and parasites was dispersed. And so around about the middle of 2010, I was contacted by this fellow who I'm sure you'll recognise, David Hume, who asked if I'd be interested in coming to the Roslin Institute. Now, this has really been a fantastic move north for me, both personally and professionally. And I initially joined as a reader in 2011 and became personal chair in microbial pathogenesis in the same year. And I've continued my interests from my days at the Institute for Animal Health and been fortunate to have a wonderful team of young researchers who have really helped to rederive all our expertise and our models. So what do we do in my laboratory? Well, I'm interested in bacterial pathogens that are both found in animals and can sometimes transmit through the food chain and the farm environment to people. We call these zoonotic pathogens. And I'm particularly interested in these three: Campylobacter, Salmonella, and E. coli. And collectively, these three organisms cause around 75,000 laboratory-confirmed cases of acute gastroenteritis in people every year. But those are only the 75,000 that we capture through national surveillance, and there are very many more of these infections that are undiagnosed in the community, perhaps because we don't go to the general practitioner. Perhaps even at the general practitioner, we don't offer up a fecal sample required for diagnosis. And even if we did, maybe we can't get a definitive diagnosis in the lab. So it's thought that perhaps 1% of the national population, some 10 times this number, are being affected every year. And that occurs, of course, at a high recurring cost to the national economy. Now, the WHO have recently estimated that on a global scale, as the principal was saying, these organisms cause a vast volume of infection, some 174 million illnesses, perhaps 80,000 deaths, and the loss of around 60 million healthy life years. So a significant global public health burden. So my lecture was titled Confronting the Microbial Menace in Our Food, and I thought I ought to just introduce these menaces in a little bit more detail. First of all, Salmonella, and I'm going to spend a little time today talking about the work we do with this organism. Most infections in warm-blooded animals are caused by a type of Salmonella called Salmonella enterica, subspecies 1, and there are many thousands of different types of this organism that express different structures on their surface. We call these serovars. And these serovars differ in their host range and the sorts of diseases that they can cause. So there are some Salmonella serovars with a very broad host range. They can infect chickens and pigs and cattle, and also, by virtue of having this capacity to colonize lots of different hosts, they can also infect us. So these are the zoonotic pathogens that come through the food chain and farm environment and infect people. But at the other end of the spectrum are also some serovars that have become specific to a given host, and these tend to cause much more severe typhoid-like disease. The bacteria grow in the blood and they grow in the visceral organs, and these infections cause a lot of death in farm animals, and in cattle can be associated with abortion. So there are good reasons to study these from the perspective of farm animal welfare. We have got some vaccines for Salmonella. There are some used in poultry, but they're not very effective, and they're somewhat specific to certain circulating strains. And it's a shifting bug in that its evolution has been punctuated by the emergence of epidemic variants and drug-resistant variants, and so it's somewhat difficult for us to keep up with. Now, the second menace that I'm going to talk a little bit about today is Campylobacter, and in the UK and many other developed countries, this is the leading cause of acute gastroenteritis. And Most people have an unpleasant infection, but they get better. It can last several days, and in rare instances, these infections are followed by severe complications. There can be reactive arthritis, but there can also be these so-called inflammatory neuropathies. These can actually result in paralysis. Often it's only transient, but the idea that you can be paralyzed after an episode of food poisoning is obviously pretty terrifying. Now, it's believed that up to 80% of human cases of Campylobacter may be attributable to the handling or consumption of contaminated poultry meat. And this poultry meat gets contaminated during the slaughter process. So the bacteria are found deep in the intestines of the chicken, and there they can grow to simply vast numbers, something like 100 million Campylobacter cells in every gram of chicken gut contents. And so you can imagine that during the slaughter process, which is rather mechanical, and the carcasses are eviscerated, you can imagine that some of that gut content can easily go onto the surface of the meat. And if then in the kitchen we handle that meat or don't cook it properly, then we can be infected as a consequence. Although there are 100 million of them in every gram of chicken gut contents, only 500 are needed to cause food poisoning in a person. So you don't need very much chicken gut contents to end up on your chicken before you have the threat of infection. And a significant threat there is. The Food Standards Agency recently estimated that around 60% of supermarket fresh whole chickens carry Campylobacter. So about 2/3 of us are taking this bug home from us when we buy a supermarket chicken. And at the moment, there are very few things we can do about that. There are no licensed vaccines and few effective control strategies. And as you'll see from a little later in my talk, the group and I are working hard to try to solve that. The final menace that I'm going to talk about today is this one, enterohemorrhagic E. coli. And there's a clue in the title here. These are E. coli that can cause hemorrhagic colitis. This is an endoscopy image of an infant with this infection, and it manifests as bloody diarrhea. So these are really unpleasant infections in themselves, but unfortunately, a subset of patients that have this go on to have even more severe complications associated with the production of one or more potent toxins. These are the so-called Shiga toxins, and they have an ability to shut down protein synthesis, and particularly do so in the kidneys. And so infections with these organisms are in fact the leading antecedent to acute pediatric renal failure in the UK and many other countries. One of the most prevalent types of this bug is E. coli 0157. Some of you with long memories may know we had a major outbreak here in Lanarkshire associated with the butchers some years ago, killed around 20 people. And these bugs are found often living without symptoms in the intestines of cattle and to a lesser extent sheep. About 5% of the national cattle herd has these bugs in the intestines. Those animals have no symptoms, but they can transmit the bugs through the food chain and farm environment to us, and we can then develop very severe infections as a consequence, particularly in infants and the elderly. So for all three of these organisms, we've been incredibly fortunate that for about 16 years, we've had the complete blueprints to begin to understand how it is that they might cause disease. So the first genome sequences began to be published for all three of these organisms around about the year 2000 or 2001. And today we have genome sequences for tens of thousands of strains of each of these pathogenic microbes, which is great. But just looking at these genomes, which in the case of Salmonella or E. coli might contain around 5,000 different genes, it's not necessarily easy for us to see which of those 5,000 genes play a role during the infection process, when, where, and at what level they're being expressed, and therefore what the impact of variation in pathogen genomes might be. We can also have trouble predicting how the encoded proteins that are determined by these 5,000 or so genes might act, and it's challenging for us to identify which ones we should target, whether using drugs or vaccines, in order to control infection. Now, one particularly important theme that I've developed in my research and I'll talk about today is how we can answer these questions about the role of bacterial genes during infection with minimal animal use. We do use farm animals in our research. I don't like to. We want to maximize the amount of information that we gain from those animals in order that we can design control strategies, but with minimal animal use. So all three of these organisms are genetically tractable, and that means that in the laboratory we can take something away from them and create a mutant strain. So we can remove the function of just one gene and then ask what effect did that have on the ability of that microbe to cause disease. And we've done that, and there's an example on this slide. Shortly after the genome sequence of Salmonella enteritidis, which caused an outbreak, a global pandemic in fact, associated with Salmonella in eggs in the 1980s and 90s. Shortly after its genome was published, we could look through it and find that there were some genes that are predicted to encode these hair-like appendages on the bacterial surface. And these were a good candidate to be involved in the colonization process because we knew from other bacteria that these hair-like structures often stick the bacteria onto host cells. So we could predict where these were encoded and knock out each one in turn. And we did that and then studied the behavior of each of those mutant strains separately in chickens. Some of them have a big effect, some have no effect. This was one that we hadn't previously recognized, played a role in colonization, and now we know it's important. But remember, there are perhaps 4,500 to 5,500 genes in these microbes, and so to do this on a gene-by-gene basis is obviously going to be hugely time and animal intensive. And so it really wouldn't be ethically responsible for us to carry on with an approach like this. We have to find another way of screening mutants for a loss of virulence in animals without doing this gene-by-gene mutagenesis. Now, don't be alarmed by the content on this slide. I'm going to walk you through it gently. This was a method that we rapidly adopted at the Institute for Animal Health to screen pools of mutants. Rather than screening one mutant at a time, here we were putting perhaps 95 together and then following the fate of every one of those during their infection in animals. Now, this is a process with a long name, signature tag mutagenesis. Don't worry about how the detail works. Suffice to say, what we do is randomly mutate the bacteria. We do this with a so-called transposon, and you can think of this rather like a jumping gene. It hops almost at random into the bacterial chromosome, and when it does so, it inactivates a gene. And we have a panel of these transposons. There are 4 of them shown here in different colors, where each has a unique barcode. This is the signature tag, and it's something that uniquely identifies the bacteria into which that transposon inserts. So what this technically allows us to do is randomly mutate the bacteria and then assemble a pool of mutants where each lacks a different character and where each has a unique barcode. Now what we can then do is take this pool of mutants, and we often have 95 at once, and give them to animals. And at some time after infection, we then ask what happened to all of those members of the inoculum. Did they survive and colonize the host, or did they fail to colonize the host? And we can do that by looking for the presence of these barcodes. These signature tags are unique to each population member, and so we can scan for them before and after infection, and in this way hope to identify mutants which could no longer colonize the animals. So there's an example here in this hypothetical experiment of 4 different tagged mutants. All of them, not surprisingly, are found in the inoculum. 3 make it through the animals and colonize successfully, but you'll see that 1 here in red does not. And we can infer from the fact that it's missing that this mutant must have a defect in a gene that's important for colonization in the host. So in this way, we could follow what happened to 95 mutants in a single animal instead of having to test one by one at a time. So it dramatically cuts the number of animals that we need to use in experiments. And at the time, this gave us so much more information about how Salmonella colonized the gut. Now, not only can you study infection at a single site and a single time using this sort of approach, but you can also follow how infection spreads within an animal. And you might remember me saying that there are some types of Salmonella that can leave the gut and cause a systemic disease where they affect the blood and they affect the organs. And here what we've done is use a surgical model where under anesthesia in cattle we can insert draining tubes into the lymphatic system or the blood supply so that we can capture Salmonella as they leave the intestines in real time and then ask questions about the role of different bacterial genes in this process. Each of these black spots here is the signal that we return for each of those barcodes. So if there's a big black spot, that mutant is present at all of the sites. If the spot disappears, then the mutant is weakened, it's missing at those sites. And so we could follow a pool of mutants where all lack a different character and watch whether they can go into the gut wall, escape to the lymphatic system, leave in cannulated lymphatic vessels or arrive at the visceral organs. And all of this in one animal. So there are 40 or so different mutations under study here, but we're following the fate of all of these mutants in one animal at once. And so that's a major, again, advance for the 3Rs in that we can get information about what all of these genes do, but from using one animal instead of 40. And this has helped us to understand why different types of Salmonella cause different types of disease. Now, again, don't be alarmed by the technical jargon on here. This is just to bring you up to the current state of the art in the way that we do this sort of analysis of microbial genomes. And this was a technique that my lab adopted just a couple of years ago to assign roles to bacterial genes with really minimal animal use. And it's by far the most sophisticated way in which we screen mutants at the moment. There's a lot of technical detail on here, but conceptually, it's just the same as the method I've already told you about. We use a transposon to randomly mutate the bacterial genome, and then we take pools of mutants, sometimes hundreds at a time now, and we can screen those pools of mutants in animals. And again, we compare what went into an animal with what comes out of it. It's just in this case, instead of looking for the presence of unique barcodes, what we instead do is use a clever sequencing strategy to sequence the flanking regions either side of the mutations. Now, this sophisticated method we call TraDIS has been incredibly powerful in assigning functions to bacterial genes in the host, but with minimal use of animals. In this hypothetical experiment, there are two genes, A and B, and they've each been mutated 3 times at these positions. And when we apply this clever sequencing strategy, we return these sequence reads either side of the transposon insertions. Now, this is giving us 2 types of information at once. We can see where every mutation is located, and because of the way the sequencing works, we can tell how abundant the mutant those sequence reads belong to is within a pool. So simultaneously, we get identity of the mutants and how abundant they are. And if we then compare this before infection of animals and after infection of animals, we can begin to see which genes are important. So there are mutations in this gene, and they're represented both in the inoculum and in the output pool from these animals. So this was a mutated gene and the mutants were screened, but this gene's not important for survival in the host. Whereas on the left-hand side, you can see another gene again mutated 3 times. Clearly that mutant is represented in the inoculum, but this time it's missing after we've screened it through animals. So simultaneously we get the identity and phenotype, the fitness of almost every mutant that we screened. And to cut a long story short, this was a major endeavor for my team and also collaborators at the University of Cambridge. We've made a comprehensive library of Salmonella mutants, and we've then screened those in cows, pigs, and chickens. And there were almost 9,000 mutants that we've screened here in these animals. And using this clever sequencing strategy, we were able to work out both the identity and the phenotype of about 90% of those mutants. So we've screened almost 8,500 mutants, but we've been able to do so with minimal animal use. In calves and pigs, we could screen 475 at once and get the phenotype and identity of about 425. So all those phenotypes just from one animal. And we annotated the function of about half the Salmonella chromosome using 18 calves and 18 pigs. So it's a really powerful strategy to get functional annotation with minimal animal use. I won't go into the detail of what we found. Suffice to say, there are many genes that play a significant role in colonisation. Some of them are important in all three hosts and play a conserved role. Others seem to be important in a cow, but not necessarily a chicken, and vice versa. And of course, this is critical information for the design of control strategies, because something designed to work against a factor in chickens might not necessarily have the same effect if you used it in another host. So this now is the substrate for a lot of the work that we're doing in the lab to really take forward this information and design effective control strategies. Now, not only are these sorts of approaches really powerful when you use them from scratch, you can also go back historically and apply them to things that you've screened before. And back when I was a postdoc at the Institute for Animal Health, we'd used that first approach of screening mutants that I mentioned to screen a library of E. coli 0157 mutants in calves. Now, that was actually a really difficult thing to do. E. coli 0157 is classified in hazard group 3, which means that to do these experiments, you had to be dressed somewhat like this with a filtered air supply. So we're not going to go back and do these sorts of experiments again with a more comprehensive approach. What we instead did is we went back to a library that we had screened and applied this much more sophisticated method to work out what was present and how did it behave. And so we could go back without using any more animals and experiments and apply this much more sophisticated method. And where back in the day when I was a postdoc, we managed to get the identity and phenotype of about 4% of the mutants that we screened without doing another animal experiment, using this much more sophisticated sequencing strategy, now we have the identity and phenotype for over 90% of what we screened. So much, much richer information. And there's just one example here. We knew this cluster of genes was really important because we'd mapped attenuating mutations that impaired the ability of E. coli to colonize the gut. But without using any more animals and screening this same library by a more sophisticated method, we realized we'd missed tens of additional attenuating mutations in this region. So it unlocked a lot of the missing information from our earlier screen. So what sorts of things did we find when we did this analysis? Well, for both Salmonella and E. coli, it turned out that they used these just remarkable molecular syringes. These are incredible nano machines that span both the inner and outer membrane of the bacterial cell. And they use these molecular syringes to colonize the intestines. Now, for Salmonella, we know that one of the things they use one of their molecular syringes for is to invade the gut. These are Salmonella bacteria here, and within just a few minutes of them coming into contact with cells lining the gut, they've caused the lining of the gut to form these ruffles, splashes of membrane, and these ruffles internalize the bacteria. Now, this isn't the normal response of a cell lining the gut to a bacterium. The cell lining the gut has been triggered to form these ruffles through the use of this molecular syringe. The bacteria poke it into cells lining the gut, pump down it a series of their proteins, and those proteins then hijack normal processes within the host cell. In this case, they dramatically rearrange material underneath the membrane to cause these dramatic ruffles. And so this molecular syringe plays a critical role in bacterial uptake from the gut. Now, bear with me on this because it looks like a rather macabre model at first, but again, this has proven to be a really powerful method this time for us to be able to quantify how Salmonella cause gastroenteritis. Now, this is a surgical model. We actually won a 3Rs prize for this. We actually use Frisian bull calves. So these are male dairy animals. They cannot make milk, and as a consequence, they are killed in early life because farmers can't afford to keep them, and they're sold into veal production. We use very small numbers of these Frisian bull calves and put them under terminal general anesthesia. So they're asleep for the whole procedure, and while they are, we can exteriorize the small intestine, which is vast in animals of this size, and we can ligate this gut into a series of discrete segments. We can maybe make 40 or more ligated segments in the bovine intestines, and we can do that by tying surgical silk around the intestine to tie off the gut lumen. Now, you can do that at this region of the intestines in cattle because the vasculature, the blood supply, is very highly branched, and so you can tie off the gut without restricting the blood supply. So these ligated loops, each about the length of my index finger, are miniature intestines, and each one of the 40 can then be inoculated with a different strain or treatment. And the animals are under terminal anesthesia for the 15 or so hours it takes us to do this. Now, if you put a wild strain of Salmonella into one of these loops about the length of my index finger, then 12 hours later, this is what comes out. This is a 50 milliliter falcon tube, and this is 30 milliliters of fluid with a lot of inflammation. And so this is modeling what happens during Salmonella gastroenteritis. In this one region of the intestine, this wild strain of Salmonella causes a lot of fluid accumulation and a lot of inflammation. If you take away that molecular syringe and put that mutated bacteria in an adjacent loop, you get absolutely nothing. No fluid, no inflammation. So that one molecular syringe is critical in determining the ability of Salmonella to produce this extreme pathology with a lot of fluid accumulation and inflammation. And implicit in the notion that the syringe is important is that what goes through that syringe must also play an important role during disease. And I and other collaborators have spent a lot of time trying to understand the contribution of some of these injected molecules, which then hijack cellular processes. And we can get all of this information from one animal, where we're taking many different strains or treatments and testing them at once for their ability to cause pathology. And actually, with this information that the needle is important, we've gone on to test small molecule inhibitors of this apparatus, vaccines based on needle components, and also to test the safety of live vaccines as well. For E. coli 0157, we also know that molecular syringes play a critical role in colonization of the gut. E. coli 0157 doesn't use its molecular syringe to invade the gut. Instead, it uses it to stick to the surface of cells lining the gut on these raised structures, and it does so very intimately. Remarkably, these bacteria use their syringe to pump their own receptor into cells, and this receptor then engages a protein on the surface of the bacterium. So they put a tube into a cell lining the gut, inject a receptor into it, and then engage with it, and that mediates this very intimate attachment that stops the bacteria from being flushed out of the gut. So without this syringe, they very quickly disappear, and Professor David Galley here at Roslin and colleagues elsewhere at Easter Bush have since gone on to show that many components of this system make effective vaccines to control E. coli 0157 in cattle. And again, we've spent a lot of effort trying to work out what goes down this needle and why do those injected molecules benefit the bacterium during infection. The last couple of things I'm going to say about how we are studying bacteria at the moment relate to how we're studying the behavior not of mutated strains this time, but also wild-type strains. Our collaborators in Zoetis, formerly Pfizer Animal Health, were very interested to know, do all of these different types of Salmonella pose an equal risk of causing disease in animals and an equal risk of contaminating parts of the animal that we eat. And we could, of course, take each one simultaneously and test it separately in an animal. But to do that would obviously be very time and animal intensive. And so what we've very recently been doing is, again, throwing them together in pools and using the power of DNA sequencing to work out what happens to them all. And we can do that because there's a part of the Salmonella genome that's very variable in sequence. And so if we throw all of these serovars together and then sequence this region, if we get a sequence read with this sort of sequence, we'd know it was this serovar. If this one, it would be this serovar. And so we can tell what's in the population, and because of the way the sequencing works, we can also tell how abundant they are. So we can put all of these together in an inoculum, and then after infection, work out what happens to them. And we've done just that only very recently, looking at this pool of mutants. So this is what went down the neck of one animal. There are 11 or so different strains here. And then we can follow, using the sequencing-based method, what happens to them all. And you can see there are some real surprises here. This one in yellow dramatically outcompetes everything else in the intestines. But what's in the intestines bears no reflection to what's in the lymphatic system. And most of the strains that we tested had the ability to leave the gut and set up home in the lymphatic system, which is a problem because these lymph nodes often end up in our ground beef and in turn can contaminate a beef burger. And now we know if we want to prevent that from happening, we need to be able to tackle all of these. It's not just one or two that pose a greater risk of contaminating these sites. So again, it's a powerful approach for the 3 Rs, in that in one animal, we could see what happened to all of these different types of Salmonella without having to take each one separately and give it to a different animal. So just for the last 5 minutes, I'm going to depart from talking about bacterial factors that are involved in colonizing the host, and instead I'm going to talk about how we identify host genes that influence the outcome of infection. Now, the Roslin Institute, of course, is very famous for studying animal genetics and identifying regions of the animal genome that associate with traits of interest. And in our case, one of the traits of interest is resistance to infection. And in particular, we'd really like to have chickens that don't have Campylobacter or Salmonella, or ideally have fewer of these bacteria in their intestines, so that there's less of a threat to food safety. Now, out here at Easterbush, we're incredibly fortunate to have the National Avian Research Facility, and within it we house unique inbred lines of chicken that heritably differ in their resistance to a range of bacterial, viral, and parasitic pathogens. And in particular, there are some that are relatively resistant or susceptible to Campylobacter. Now, this is a fantastic resource because what we can do is we can mate those chickens together that are either resistant or susceptible. And obviously, the progeny of that cross will inherit genes from mum and dad. And depending which ones they inherit, they could become more or less resistant or susceptible to infection. So what we can do is sample genetic variation at tens of thousands of positions along the genome of these birds. And we do that at specific positions, single nucleotides at tens of thousands of positions through the genome. And we can then build up a picture of which variation is associated with resistance to infection. So we can challenge these birds, look at how they respond to infection, and simultaneously look at their genetic makeup and begin to associate statistically their genetic makeup with their response to infection. Now, I mentioned that we've got 2 inbred lines that differ very greatly in their resistance to infection. Here they are in their response to Campylobacter. One line gets colonized and the other very poorly gets colonized. Now, you'd think, well, fantastic, problem solved. Why can't we just eat these guys? But unfortunately, these are inbred layer lines. They're not meat birds, and they'd be absolutely useless in a commercial setting. They've not been selected for the meat that they produce. So we can't just eat these inbred layer lines, they wouldn't perform as a commercial broiler bird. But nevertheless, these lines are a very powerful resource for us to understand the basis of resistance to infection. And I won't go into the details of how we cross them, but we've made crosses of these lines and then begun to associate within the progeny of those crosses their response to Campylobacter with their genetic makeup. And what you end up with after a lot of hard work is something like this. It's called the Manhattan plot, and I'm not going to go into the detail, but at some positions within the chicken genome, you go above a magic threshold where over this line there is a statistical link between variation at these positions in the chicken genome and resistance to Campylobacter. So of all the positions that we sampled, the vast majority have nothing to do with Campylobacter resistance. But at some positions, we see that there is a link between variation there and having a more resistant bird. And that's potentially very useful information for the design of control strategies, because we can ask breeders to select for it in their breeding schemes. We've identified a few regions of this kind, and it was published earlier last year. Now, doing this sort of thing in a layer line is all very well, but of course you worry about whether or not that information really is relevant to a modern commercial broiler bird. And working together with Aviagen, there was a major genome-wide association study performed here in the last few years, where we have many thousands of commercial broiler birds that were exposed to Campylobacter out there in the real world. And we've got high quality information on their genetic makeup and high quality information on how much Campylobacter they had. And we can statistically link the two. Where is the genetic variation that associates with being resistant to infection? And again, we found regions of the chicken chromosome that appear to determine resistance. And some of these, remarkably, are the same as those that we found in our earlier study using the inbred birds. So there really is a heritable component to resistance of chickens to Campylobacter, and I believe it's worth trying to select for that in modern poultry production. There's a relatively modest contribution of genetics to the phenotype, but I think it might be part of a complex solution to control of this bug. Now, I really can't mention this study without paying tribute to a dear friend and former colleague of mine, Professor Peter Kaiser, who was a professor here at the Roslin Institute and stood on this stage not so long ago. And as well as being a great friend, Pete and I had many productive collaborations, not just on Campylobacter, and it was Pete that initiated this study. We're working hard to try to bring it to fruition. We also work with him on many other projects in Salmonella and pathogenic E. coli. I'd really like to pay tribute as well to my former team at the Institute for Animal Health. I was very fortunate to work with some talented early career researchers there. It was a great pity that my well-functioning team was dispersed through a process of redundancy, and some of them left science as a consequence. And it's really them that drove my research profile in those 12 years or so that I was at the Institute for Animal Health Compton Laboratory. But I've been so fortunate to come here to the Roslin Institute and have a wonderful new team here that have been fantastic in rederiving our models of infection in farm animals and our expertise. I really couldn't get back to a competitive position without all that they've invested on our behalf. I owe them a great debt, and I'm sorry if not everybody here from the Roslin Institute is named, but these are some of the folk that we've been collaborating in particular with lately. Of course, behind all of this is a great deal of support from my family, and without their love and encouragement, I really don't think I'd be half the scientist. My dad, Peter, here, my mum, Sue, on the end, my sister, Sharon, and her young family, and of course my wife, Jo, and lovely son, Ben, who put up with a lot from me in terms of late work and working at weekends, and I really think I owe a lot of my success to the support I've had from them. So I'm happy to take any questions from the audience, and thank you for your attention.
Thank you very much, Mark. The principal has just asked me to do the vote of thanks, and I'd like to be the first to congratulate you on your inaugural lecture. That was absolutely excellent. I'd just like to reflect on a couple of things. I think that we've learnt a lot from your lecture. We've learnt that your fashion sense has come a long way since 1988. I now know why you're late to work, because you're still travelling from Warwick to the Roslin Institute every morning. But more importantly, and jokes aside, I think we've learnt the importance of mentorship. I think that was really important. The importance of bringing on the next generation. I thought your work is really incredible. It crosses the species boundary. It's absolutely what this college is about, the one medicine principle. And I think finally you can demonstrate clear lines to impact. This is going to really affect lives both in animal health and in human health. So I would hope that everybody can join me again in just thanking Mark for a wonderful inaugural lecture.